Good evening everyone, welcome back to my channel. In my last video, I have shared glycolysis, which is a very important topic for CSIR NET, GET and any competitive examinations. So I have randomly picked two questions for today's discussion. That is, one is from GET 2025. That is, which of the following reactions is or are catalyzed by aldolase? We all know aldolase is an enzyme, right, which is used in glycolysis. And the options: dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate to fructose 1,6-bisphosphate. First option is that. Second option, dihydroxyacetone phosphate plus erythrose 4-phosphate to pseudoheptulose 1,7-bisphosphate. Option C, dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, and option D is glyceraldehyde 3-phosphate plus erythrose 4-phosphate to pseudoheptulose 1,7-bisphosphate. So, is there any, can you guess the correct option? Yes. The correct option is option A, that is dihydroxyacetone phosphate. Dihydroxyacetone phosphate is also having a ketone group DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate, that is G3P, glyceraldehyde 3-phosphate, right? So we have seen in glycolysis that fructose 1,6-bisphosphate in presence of this enzyme aldolase splitted into two 3-carbon compound. One is dihydroxyacetone phosphate and the other one is glyceraldehyde 3-phosphate. And within these two compound, they are isomer to each other because they are three carbon compound and they can transfer into each other by the enzyme phosphotriose isomerase, right? So go and check that video so that you can get the correct option. And so the correct option is fructose 1,6-bisphosphate. It can split into these two 3-carbon compound by the help of aldolase. Okay, and there is one other correct option, that is option B. Option A and option B both are correct because option B is also true for aldolase. Aldolase for dihydroxyacetone phosphate plus erythrose 4-phosphate, these two conversion to pseudoheptulose 1,7-bisphosphate, this is true for Kelvin cycle, and which is for plant, and for animal that is glycolysis, option A. Okay, so option A is glycolysis. Option B for Kelvin cycle. Kelvin cycle for plants and glycolysis for animals. Okay. My next question from GET 2024, that is, which one of the following metabolic intermediates is common for glycolysis, nucleotide synthesis and glycogen synthesis? That means choose one compound which is common for these three type of biochemical pathway. One is glycolysis, nucleotide synthesis and glycogen synthesis. Option is A citrate, B oxaloacetate, C glucose 6-phosphate and D glycerol 3-phosphate. Option A, obviously I know that glucose 6-phosphate. Again from glycolysis, this is the important compound for glycolysis, nucleotide synthesis and glycogen synthesis. Why these things are not? Because citrate is from TCA cycle, oxaloacetate is also from TCA cycle, intermediate of TCA cycle, these two, and this one glycerol 3-phosphate is for lipid synthesis. That's why these are not correct. The correct option is glucose 6 phosphate. So stay tuned with me for getting this type of previous year question, and I will like to share the importance with you also. Bye bye.